Assalamualaikum warahmatullahi wabarakatuh. My name is Faris Nulhaqim bin Muhamad, and my metric number is 200663. And today I'm going to show you a simple experiment in practical 6, which is about alcoholic fermentation in yeast. As a rough introduction, alcoholic fermentation, or also known as ethanol fermentation, is an anaerobic pathway which is done by yeast, which the yeast converted simple sugars into ethanol and carbon dioxide. So basically, yeast uses anaerobic pathway to gain energy in the form of adenosine triphosphate (ATP), and the byproducts excreted is ethanol and also carbon dioxide. The application of alcoholic fermentation in yeast is widely used in our daily life, such as the production of wine and also the making of bread. The experiment is actually very simple, in which it only requires a few materials and substances which can also be obtained in our house. So basically, first of all, we need 4 bottles. We also need labeling, in which I have already labeled it. I will explain it later. We also need yeast and the carbohydrate sample, in which I use starch, which is the rice flour, table sugars, and also glucose. We also need tablespoons to measure, and also balloons to indicate how much the yeast excrete out carbon dioxide. And we also need warm water, which the temperature is approximately 37 degrees Celsius. So first of all, I'm going to explain you about the ingredients in each bottle. So the first bottle, I'm going to put water and yeast, and also starch as the glucose, as the carbohydrate sample. In the second water bottle, I'm going to put yeast and water too, but this time I'm going to put glucose as the carbohydrate sample. And the third bottle, the same, I'm going to put water and yeast, but this time table sugars as the carbohydrate sample. And the last one, I'm going only to put yeast and water, but no carbohydrates, which it will act as control in this experiment, while this 3 is experimental group. So I actually use a different type of bottle because my recording just stopped when I was doing with the other bottles. So I prepared these 4 bottles, and basically what we are gonna put in these 4 bottles is 20 milliliter of water, warm water, and carbohydrate sample. First bottle starch, second bottle glucose, third bottle table sugars, and fourth bottle no carbohydrate sample at all. And also in all of these 4 bottles we are gonna put yeast in it, which is 1 tablespoon. The carbohydrate sample we're gonna put also 1 tablespoon in these 3 bottles. So in the first bottle, $20 milliliter of warm water will go in it. One tablespoon of starch, in which I will use rice flour. and then 1 tablespoon of yeast. Then we're gonna mix it up well. After that, we are going to put a balloon on top of it to measure the size of it, to indicate as an indicator of carbon dioxide being excreted out by the yeast. So we are going to take up to 25 minutes of observation, with 5 minutes with each 5 minutes we are going to take a look at it. Now we are going to do the same thing on all of these 3 bottles as well. Do the same thing on the second bottle, except for the difference between the first bottle is that I'm going to put glucose as the carbohydrate sample instead of starch. Bottle, the same thing, except the carbohydrate sample is table sugar. And the last one is the control. in which I will only put yeast and water in it, but no carbohydrate sample.